Urbana-Champaign. He did a master's at the University of Missouri and went back to UC for a PhD with Dr. Okay. He then did postdoctoral work in Dallas with Joel Elmquist at UT Southwestern, and then at UT Dallas with Dr. Ted Price before starting as assistant professor in 2017. Now, Dr. Burton's a certifiable rising star. He actually won an award that says he's a rising star and has won multiple K-level awards and other early career fellowships. And I can see why. Dr. Burton works at the interface between the nervous system and the immune system and has made fundamental mechanistic discoveries about how these dazzling biological systems work together. The title of this talk is The Cell and Sex-Specific Role of TLR4 in Mediating Inflammatory Pain. Thank you very much, Dr. Burton.
Awesome. Thank you so much for that lovely introduction and for the invitation. So I can come out and share a little bit about what my life has been doing for the past going on 5.5 years now. This has been a long time coming. You just reminded me, I've been wanting to visit Iowa for a very long time. So really happy to be here. All right, so let's jump into it. I know we got a little bit of time there. We can do that. My lab opened at the University of Texas at Dallas. This is Richardson, Texas, actually, which is a north suburb of Texas. It's of Dallas, and it's we call it UT Dallas. Is super interesting. But this beautiful building opened up in 2016, and my lab moved into the basement down where you don't have any windows, essentially over here, but still a beautiful place. Make life a little bit more simple. And I like to show this picture. This is like my second lab meeting that I ever had. And I really want to talk about and really focus on how we really want to engage training at all levels, undergraduate, graduate, post-bac, postdoc, have some medical interns as well in this space, and just to engage as many people as possible at all different levels for just educational diversity, internal diversity, et cetera. We're a part of the Center for Advanced Pain Studies, which is a fairly new center that I'm a co-founding member of as of 2019, along with Ted Price and Greg Dusor, where we have funding from the University of Texas system, as well as other funding sources as well. For me, I always like to tell people a little bit about where I come from because it's kind of diverse. Where my training and expertise is kind of three different things that all kind of came together to this kind of beautiful symphony, if you will. I was kind of apprehensive about putting the Illinois brand on there, but that's where I'm from. I was actually born and raised in Chicago. I went to the University of Illinois for my PhD, where the focus of that was immunophysiology and behavior. So understanding how the immune system interacts with the brain and nervous system to facilitate behaviors. Moved on to UT Southwestern to work with Joel Elmquist, who the major goal or focus was endocrinology and metabolism with a hardcore focus on molecular genetics and neuroanatomy. Projects went different ways and interest kind of waned, and I did something that people typically don't do during your postdoc. I did a second one in a completely different direction, looking at pain. Just so happened that we had a really interesting cell type phenomenon that my first postdoc mentor had no interest in whatsoever. So I had to find a second person that was really interested in. If you ever met Ted Price, he's really interested in everything and happy about engaging as many people as possible. So it was a really nice thing. It's only about a 17-mile difference between the two institutions, and driving between them can be really, really fast, like 20 minutes or as far as an hour and a half to get to the two different institutions. But my lab focuses on integrating the approaches of understanding how lifestyle factors, age, diet, ethanol consumption, and how they interact with these discrete systems. And it's the communication between the nervous system and I guess that's missing, and influence between the communication between the nervous system and the immune system, these discrete systems. And so I always like to talk a little bit about my lab philosophy, just to kind of show where we come from. And There's kind of three major points, which is everyone has access to all information and resources to make complete and educated decisions. And to be completely deliberate in your actions and whatever that may be, especially with trainees at all different levels, and making sure that everybody's empowered to be the best they can be. And understand at the very bottom here, good science is for everyone. It should be shared 100%. And it's something that we really stand by in the lab space. So the lab has four projects. I'll talk about one at all four. And the first big one that I've been kind of obsessed with for a long time is aging. Just because that's the one thing that everybody hopefully will have the experience of getting older. And it becomes more and more apparent as I get older how important it is. It doesn't seem to be that important when you're a little bit younger, but when you wake up with the backaches and the vision issues, it starts to become super relevant. So how aging and sex influence the immune system, metabolism, and other behavioral phenotypes. The second project is just like nitty gritty neuroimmune communication and two different components. One is cell-specific actions of TLR4. The other one is looking at different types of chronic muscle pain. And so the first, I didn't mention, the first project is we have aging animal models and clinical data and some patient data. The second project is also animal models and patients. The third project is the role of cannabinoids mediating affective behavior in analgesia, which is pretty much exclusively a novel animal model. And then the last project is actually neurosurgery. We're looking at neurosurgery patients and the role of neuroimmune biomarkers that are indicative of perioperative pain and delirium as well. And so, but like I said, we were only going to talk about the second project, but I always tell people about all of them so that if you're interested, you can email me and we can have discussions about whatever you'd like, because we can keep going. All right, so this is no shock to folks of the problem that we like to talk about or think about, or I guess focus on in the lab, which is pain, and it affecting more than 1 billion people worldwide. 1 out of 5 people suffer from chronic pain or some kind of chronic pain. Interestingly enough, there is a disproportionate amount of women that and chronic pain states that are disproportionately women, and with annual cost of over $600 billion a year in the context of lost wages, therapies, alternatives, healthcare. Interestingly enough, over about 105,000 people died in 2021 from opioid overdose in the context of the opioid epidemic. Interestingly, in 2005, it was closer to 57,000. And honestly, during COVID, it was like, well, maybe things will change and be a little bit different. And it's just compounded and keeps going. And so that brought the question if we can really discover and think about efficacious non-opioid based therapeutics, alternatives, conjunctives, however you want to say it, just something a little bit different to use. And importantly for us in my lab, we're really interested in how mechanisms of pain signaling differ between sexes and why there's more disproportionate amounts in different sexes. The sources of pain that we study in my lab is kind of a lot, kind of diverse. We look at acute inflammatory models for local and peripheral actions, long-term inflammatory pain models, chemotherapy, induced models, diabetic neuropathy models, as well as surgery as well. And looking at the intersection between social factors, genetic factors, biophysical factors to think about the experience in itself, and kind of approaching it from the molecular translationally up into the clinic. And so the neural immune involvement from my perspective. So the idea of how various immune cells interact with peripheral sensory neurons, we'll say nociceptors in the periphery, it's been vast and been quite well described. You have cytokines, you have inflammatory factors that are released and interact at nerve endings to cause sensitization and changes in various ion channels that lead to differences in pain thresholds. Now, what drove my postdoc kind of shift was we had an animal where we're looking at sensory neurons, but in the context of metabolism. And we didn't see much of a metabolic effect or a whole body metabolism effect, but we were really interested because the same neurons that we're looking at were sensory neuron in the context of pain. And this paper from Clifford Wolf's lab was published in 2013, I think, ahead of print, where they showed and described how bacteria activated sensory neurons directly, as opposed to the prototypical thought that a bacteria would come in, interact with an immune cell. The immune cell would then release all these particular cytokines and factors and then synthesize sensory neurons. And so down here is looking at how they tagged some peripheral sensory neurons within the skin. And then they actually did a dual tag with Staph aureus, and they can see directly that there is some interactions. And with the sensory neurons, but also obviously plenty of other cells within the skin. And then if you looked a little closer, flip the colors, you can still see that the bacteria were directly interacting with the sensory neurons as opposed to the immune cells. And this particular, I guess, graph kind of really hit it home for me, which was looking at how usually when you have inflammation in the context of bacteria, that's associated with some type of pain. But previously, I guess it was thought that tissue swelling and the infiltration of the immune cells was driving the pain. But if they took a really, really close look, they saw that bacteria unload and pain were pretty much overlapping, and that tissue swelling that in immune cell numbers actually was a little bit later in effect. Meaning it was like, okay, well, the bacteria themselves are actually driving the pain with with interacting with the sensory neurons. And like I was saying before, we were really curious about the time it was like foodstuffs and how they interact with sensory neurons. And then this paper came out and showed that there is this really missing slide, really good pathway that showed that peripheral sensory neurons express things that you find in the immune system, like toll-like receptors, and also there's something called FPR1. And that kind of set it off for me. And I was like, I really want to do this. This is really interesting. These systems are discrete, but so not at all. There's so much overlap. It's ridiculous. It's so ridiculous that what you thought was an actual immune cell is actually on a sensory neuron driving pain. And so what the approach really was to understand pain plasticity pathways involved in acute to chronic pain, to identify targetable cell types and neuroimmune pathways that mediate these phases of pain, and really understand how these cell types mediate sexual dimorphic actions. And right around that time, 2013, 2015, there was a couple papers that were super huge in the field. One was in Nature Neuroscience, and this was 2015, where it showed, or they spoke on how different immune cells mediate mechanical pain in males and females in very different fashions. This was Jeff Mogul's lab. And a couple years before, they were showing how toll-like receptor 4 mediated sexual dimorphic effects as well of pain states and pain outcomes. And it really kind of got us thinking about the model that we had, which I'm going to talk about in just a little bit. So I always have to talk a little bit about TLR4 first. What is it exactly? Well, and where is it found? And it's important because a lot of early experiments use whole body knockouts for TLR4, and that helps, but it doesn't give you exact, I guess, resolution in the context of what cell types that are very important. It's found on macrophages, microglia, peripheral neurons, stromal cells, epithelial cells, more. And it's a pattern recognition receptor that binds both pathogen-associated molecular patterns, like what you find on the cell wall of gram-negative bacteria, but also binds to danger-associated molecular patterns. And a big one, which we'll talk about a little bit, is high mobility group box 1, as well as heat shock proteins. And so what we wanted to do was employ a way where we can target specific cell types like peripheral sensory neurons, microglia, stromal cells, or fibroblasts, so that we can kind of parse out its role on those particular cell types. I teach a neuroimmunology class, and the biggest kind of concept is, even though this particular receptor is on this cell, depending on what cell it's on, is going to do something just discretely different enough. And that outcome could really change a lot, right? It could beget inflammation, it can cause sensitization, et cetera. Okay, so strategies. And so what we decided to do was think about toll-like receptor 4 and just the pathway of pain. And so we're interested in nociceptors and interested in one of the primary cell types that express toll-like receptor 4, which is macrophages, and split it up. What is the requirement of toll-like receptor 4 on these particular cell types to mediate the onset of pain state, to mediate plasticity machinery, and we'll kind of go into the development of long-term chronic pain. And then the second half of the project, and the more cumbersome, bigger half, is the sufficiency aspect, which is if you take TLR4 away from the entire body and re-express it only in a specific cell type, what does that do? Is it sufficient enough to drive the wild type phenotype or recapitulate the wild type phenotype? So we took these two approaches to try to understand what TLR4, its role in various cell types. And so I was saying earlier that this danger-associated molecular pattern, HMGB1, is a TLR4 agonist, and it binds TLR4 in a redox-dependent fashion. And so we utilize something called disulfide HMGB1 to specifically target toll-like receptor 4. And the reason why that's interesting and important is because what ends up happening is in the context of activation in the peripheral of the cell, HMGB1 actually lives inside of the cell within the nucleus, and then it ends up kind of leaking out and being released and being activated after some time. And so to look for it and to use it is very important in the context of redox and in the context of phase. And so we went ahead and at the time, and we'll talk about this too, had a really great collaboration where we can get this disulfide HMGB1 because it needed to be processed properly. And we injected it plantarly into mouse paws and wanted to understand the kind of progression of pain. And so, and this is in the acute phase. So we did this dose-dependent dose response test, where in males and females we gave different levels of HMGB1. And what we found is a dose-dependent effect. The higher the amounts, the more pain that they're in. This is Paul withdrawal threshold. The lower you are on the scale, the more pain you're in. You start off at a clean 1 gram with a mouse, and the lower you go, that means they're responsive at a quicker or a more lower threshold. And so we can see that in about 24 hours, both males and females are fairly sensitive, but they end up going back up to fairly normal at about 2 days, 3 days. And our interest in translational machinery is trying to understand what happens when you have the animals at baseline and then you give them something called prostaglandin E2 at a subthreshold dose. And that subthreshold dose, when you give it to an animal that didn't have a previous pain state, you end up getting really no effect because the thresholds haven't changed. That pain plasticity machinery, say that 4 times fast, isn't as in effect. However, in a dose-dependent fashion, we can see that even though the animals are at baseline and they're giving a subthreshold dose, that you see that they are sensitive, both males and females, fairly the same. And so that gave us an indication that the HMGB1 is causing this acute effect, but is also causing a change in pain plasticity machinery to kind of that chronic or transition to chronic effect. And then we were looking at paw edema, and we wanted to just see how much inflammation was there when you treated these animals across time. And we saw within a 4-hour period of time that there was an increase, a dose-dependent increase and paw edema as well, which was what we kind of expected. And so whenever I talk about this transition, I always have to bring up John Levine's work, where he essentially pioneered this idea of if you have a normal paw in a normal situation and you treat with a particular inflammatory molecule, you get an acute effect and it goes away and everybody's happy. However, if you had an event like down here where you have a priming of that paw, meaning you had an event that was previous, you were in some pain, went back down to normal, and then you came back in with the same dose, that you get this elevated and protracted effect. And so, and that's very complex, it's peripheral, it's central. And so we really were interested to understand kind of the role in this machinery. Okay, so the requirement jump in. And so what we decided to do was do this TLR4 knockout experiment. And this is the gene targeting strategy that we did when I was a postdoc. My very first project was working with another talented postdoc who's actually now at UT Dallas, where she targeted the toll-like receptor 4 gene with these LOXP sites so that when you crossed it with a particular Cree driver, you could remove it from cell types, whatever your cell types you want. And so we use these two particular cell types, NAV1.8 Cree, which is for peripheral sensory neurons, and lysozyme M Cree, which is in primarily in macrophages, but also some other interesting cells of interest. And we wanted to look at how if you remove TLR4 in the dorsal root ganglia or in sensory neurons versus in the immune cells, what happens? And so down here is an in situ of this fact, looking at the Phlox version versus the Cree plus Phlox version. And what it looks like when we put it into a graph, about 30% of the neurons in the dorsal root ganglia express toll-like receptor 4. We found this, other people have also found this as well. And then when we crossed it with NAV1.8 Cree, we see a decrease. Same thing that happens in lysozyme M Cree. This has been published, looking at gene expression of peripheral immune cells. And so the first experiment we did was kind of go back to that same HMGB1 interplantar experiment, where we did this in the animals that did not have TLR4 in the macrophages. And so to orient everybody, there's vehicle groups that are black and gray, and they're at the top. And throughout the whole course of the experiment, they pretty much stayed at the same level at baseline. And when we added toll-like receptor 4 agonist HMGB1 to the pool, we saw a similar effect where that lasted about 24 hours, went back up to baseline in about 2-3 days, and then came back in. And this is a 2-week period post this 48-hour, giving them this subthreshold dose of prostaglandin D2. And we find that those animals do in fact prime or have that plasticity machinery. However, when we remove toll-like receptor 4 from the macrophages, what we find is kind of a mitigation of the effect acutely, and then the animals didn't prime at all to that subthreshold effect, to the same dose of HMGB1, which was that middle dose on the dose response curve. Interestingly enough, we did see this little effect here at the first hour, which I'll talk about in a little bit. And these are in males, and then in females, though, when we did the same set of experiments, really the important groups with the blue and the red, we saw that independent if TLR4 was on the peripheral macrophages or not, that those females did also sensitize and then had that priming effect a couple weeks later. And so we was like, okay, well, let's flip the script and remove TLR4 from peripheral sensory. And if we do that in males down here, we see a similar effect. It's mitigated, but it's not completely gone. However, in the females, what we saw is the opposite. Is they didn't have that acute effect, nor did they have the priming effect. So we're seeing in males, peripheral macrophages are important for not just the acute part, but the also priming part or chronicity part, transition to chronicity part. Versus in females, the peripheral sensory neurons are really what's driving that acute and the acute to chronic pain aspect. What I was saying earlier is that within that first hour, you can kind of see some action happening in this particular model. We think it's because sensory neurons do play an effect, because here TLR4 is in pretty much every other cell but macrophages. So there's still, everybody's in the pool, but people are playing a little bit differently at different times. And so there's way more data there, but I wanted to jump into kind of the next part, which is the sufficiency part, which we have been really building up across the past few years. And so, and to remind folks, that sufficiency part is removing toll-like receptor 4 from everywhere and then putting it in specific places, which is way more fun. And so we utilize the same cell types, NAV 1.8 for sensory neurons, and then lysine Cree for peripheral macrophages. But this time, as opposed to floxing out the gene, we had a transcription blocker, LOXP transcription blocker that was placed in the middle, similar to if you use TD tomato, where it's in there, but then Cree-mediated recombination removes the block and then kind of turns everything back on like a light switch in that particular cell type. And so we did this in both sensory neurons and in macrophages. And this is a little bit different approach. We did some gene expression, pulled out some DRG at various levels that are relevant for pain. And we saw that in the wild type animals, you have a particular gene expression and phenotype, or gene expression phenotype. And in the TB or whole body knockouts, you have amelioration of that. And we did it in both males and females. That's not happening where that's significant. We were interested in that because that's a question I get all the time. Is TLR4 different in males and females? What's really going on? And I guess I should put that in there now, that we don't see huge differences in expression patterns in the cell types, actually, and in different places. But the model itself to remove TLR4 or knock it down does work in the absence of Cree. And then in the presence of Cree, you can find that there's reactivation of the TLR4 mRNA in particular cell types and NAV 1.8 cell types. And here goes a more fun approach, which we did with protein expression or flow cytometry. We took those same DRGs or same peripheral sensory neuron cell bodies, sorted them out, and we wanted to look at the expression pattern of toll-like receptor 4. And so really the kind of important parts are over here, looking at the wild type versus the reactivated versus the knockout. And looking for new N or for neurons, what we found is that in the wild type animals, there's about close to 30% of the neurons that express protein. When you reactivate that particular sensory neuron population, you see close to the same amount. And then in whole body knockouts, you don't see those differences. And this came from a paper that we published a couple of years ago where we're looking at neuropathic pain. And so we're looking at like how neuropathic changed toll-like receptor 4 and all these fancy things. But really the whole purpose is when we look at neurons and look for the reactivation, we see it in proteins. And then when we look in peripheral, the same cell, I guess, the same animals, so the same NAV animals, and we look for TLR4 in the sensory neuron, the peripheral macrophages, we find that it's completely decreased and it doesn't really change. And that's because the NAV doesn't hit the peripheral macrophages. And so you have that low versus in the sensory neurons, it stays high. And so that particular animal, we decided that we wanted to do a similar experiment, but this takes a lot of time. And so across time, things change. Lots change, people change, life changes. And so what ended up happening is we had this disulfide HMGB1 that was a TLR4 agonist that dried up. Our collaborator didn't have any more. The source of it kind of disappeared. And so we ended up switching over to using LPS, which is also a TLR4 agonist. We use a purified LPS that was more specific, or as specific as possible as we could find. And so with that switch, it kind of changes the narrative just a little bit, but the idea is the same where if we inject LPS intraplantarly and then look across time, what happens with the pain state. And so the difference here is that the black here at the top is whole body knockouts. This is reactivated in the peripheral sensory neurons only, and this is wild type animals. Everybody gets TLR4 agonists here. Everybody gets LPS here, because if I would have added the other 3 lines for the vehicle, it's just a bunch of, it's just kind of messy. And so the whole body knockouts, when we gave LPS into the intraplantarly, what we found is that it didn't respond. These animals didn't respond. And these are male animals that have peripheral TLR4 missing everywhere. And then when you put it back into peripheral sensory neurons, CLR4, it actually causes that acute effect that we saw initially when we removed TLR4 from the macrophages. However, the animals, the response quickly goes back up to essentially the same as the whole body knockouts, as opposed to the wild type animals, we see this effect that lasts for about a day. Doing the same experiment with females, so putting the TLR4 back in female sensory neurons, we see a complete recapitulation or sufficiency to drive that acute phase in both the reactivated females and also compared to the wild type females down here. And the whole body knockouts didn't have a huge difference. There's always that little change at the beginning. We inject it intraplantally, but in general, it's a pretty flat line. So that's sensory neurons. Kind of flipping over to macrophages now, what we find is that when you reactivate or specifically re-express TLR4 and peripheral macrophages, you find that you compare them to wild type males, that it looks pretty much right on par. Interestingly enough, at that hour, when we expect actually peripheral sensory neurons to have a little bit more of effect, you don't see as robust of an effect in the animals that have TLR4 only in the macrophages in males. And then, to no surprise, kind of at first it was a huge surprise, not gonna lie, when you put TLR4 back in peripheral immune cells in females, they didn't have anywhere near of an effect as the males. And so kind of showing both, it depends on where you are, right? So at the beginning of the story, we said, put HMGB1 or a TLR4 agonist in the paw, see what happens. Males, females, there really wasn't a huge difference between the two groups. However, when we start parsing out the cell types, if you put it on a peripheral immune cell versus peripheral sensory neurons, you see these differential sexual dimorphic effects, which are super interesting and hoping to have us try to think about therapeutic strategies and ways to employ antagonists in a more effective way. Yeah, absolutely. Donna? Yeah, yeah. So there's a group, Clifford Wolf's group did that study that showed that after, well, I guess it depends on the agonist, but after some kind of, oh, absolutely, yeah. So the question was, what's the difference, or is there a difference with the macrophages that are infiltrating and coming into the paw after the event or after the exposure versus the local macrophages? And the long story short is absolutely. So what's happening is there's infiltration of peripheral monocytes that are a little bit different than the local macrophages that are hanging out. We've done both, looking at the peripheral monocytes that are just kind of hanging out, and the local macrophages. They have TLR4 knocked out in both populations if they're whole body knockouts, and TLR4 in them if they were. And so, but I guess a really interesting question that we don't know the answer to, that now I think about a little bit, is what's that kinetics of swelling, and how does it superimpose onto what we're finding with the pain? Yeah. No, that's a good question. That we didn't ask that directly, but we could probably look at some data and parse it out though. Yeah, thank you. Yeah. And okay, so then this particular experiment is super important because we submitted this paper, and then someone came back and said, well, ISM-CRE hits more than just macrophages. It hits monocytes, it hits some granulocytes that are local. So how do you know that it's your macrophages that are really doing what you say that they're doing? And we say, it's true. We've been saying it, but we didn't really do anything to prove that it was. And so we use something called clodronate, which is liposome loaded. So it's a particular drug that essentially kills macrophages when they phagocytose them. And we took our animals and said, all right, well, if we're going to do this experiment, let's do it kind of big. And so we took both the NaV1.8 CRE animals and the LysM CRE animals and bred them together so that we have an animal that has TLR4 both in the peripheral sensory neurons and in the macrophages. And then gave everybody this drug to kill the macrophages so that we could potentially see what happens, right? Because I was worried that, well, if there's no effects, people will say, well, it could be a genetic thing or a germline thing. But if we can see that in one sex, the peripheral sensory neuron still driving effects versus taking away the macrophages, we could kind of do this in a couple different ways. And so how it worked is acclimation, did a baseline and did some injections, IV, so kind of all over the place to get rid of the macrophages. And then we did some initial experiments just to show that when we did that, the macrophages were depleted. And then we did some behavioral assessments with intraplanetary TLR4 agonists across time to see what's going on. We did it acutely, and then we waited about a week to see what happens with this priming effect. And so this first experiment is just showing what happens when we look in the paw skin looking for macrophages. And what we found is that with the clogenate, that macrophage marker that's not on here, I should say, is CD11B, is gone. And then we also looked in the peripheral blood that when clogenate was on board, that you got a decrease in the signal as well. So it's coming in and getting rid of the macrophages. And so these experiments, again, have like 16 different actual lines, but I'm only showing 3. All right, so how this worked is you got, or the animals got clogenate in vehicle, just to show that it's not really doing anything across time. The clogenate plus the LPS, so getting rid of the macrophages only in LPS, and then just no clogenate loaded liposomes in LPS to just kind of make sure that we're keeping that wild type normal, well, seeing if we're recapitulating pain. Because technically these animals aren't wild type, they only have TLR4 and peripheral sensory neurons and then macrophages. And so when you, in males, when you get rid of the clogenate, you get rid of the effect, or when you get rid of the, when you get rid of the macrophages with clogenate, you get rid of that effect. However, getting rid of the clogenate or macrophages in females did not do that at all. So depleting macrophages in females in these NAV lysosome double mutant animals did not completely change the effect. So macrophages or specific deletion of macrophages in females is what's driving this acute effect. It's a lot of folks in the pool there, I know. And then so looking at the transition phase, we waited a week and then we did the same experiment that we did earlier, which was inject with prostaglandin 2 and to see if activating or removing the macrophages was enough to elicit this priming effect. So if you get rid of macrophages at the beginning of the experiment and then do this priming effect, there is a significant difference from just the vehicle animals in males, but in females, it didn't matter, right? So if the macrophages are there or not, there's hyperalgesic priming or there's priming that's happening. There is this machinery that's turned on that facilitates acute to chronic pain states, and immune cells aren't really, at least immune cells in the context of TLR4, aren't really leading to that pain state in females versus in males, that's the case. And I always take a pause there because I'm always like, oh, I get this question all the time. Why? Why would that be the case? And the short answer is no clue, right? And that's actually what we're spending a lot of energy on right now, is to try to figure out why that's the case. I get asked the question of, so I'm kind of trying to beat it, evolutionarily, why would that be the case, and what have you? And we don't know. We have some ideas, and I can share that with you later if you really want to know. But right now, we don't know. But okay, so then the last little bit here, last few minutes that we have, I was going to talk about kind of this machinery that's involved in sex differences and toll-like receptor 4 activation, and some projects that I've been involved in. And so Greg Dusor at the University of Texas at Dallas had a project that looked and saw how CGRP that's released from peripheral sensory neurons has a female-driven effect, not specific or only, but driven. At low doses, CGRP causes pain states, migraine in a migraine model in females. CGRP does drive pain states in a migraine model in males, but just at a much higher dose. And then I was working with a good friend of mine at UT Southwestern that we're looking and seeing if you activate TLR4 on peripheral sensory neurons, you get CGRP, right? Because could that be one of the reasons that are discerning female-specific differences is the CGRP that's getting released from the peripheral sensory neurons. And so we took some wild-type animals. Animals were, they're whole body knockouts, and then animals where they're only peripheral TLR4 and the sensory neurons. Took out the DRGs and some peritoneal macrophages because we wanted to get enough of them, and put them all in the pool together. And so you have kind of this co-culture. Waited for a few days and then did some direct LPS activity. And after about 3 hours, collected supernatant with this 250 nanogram dose of LPS, and then ran an ELISA for both CGRP, and we did IL-6. IL-6 is a pretty potent TLR4 driven cytokine as well. And so this is kind of what a culture looks like, everybody in the pool. And the first kind of experiment at the top is looking at IL-6. So if we wait even longer, 24 hours afterwards, and then we take some more supernatant, what we find is if CRE is present, or CRE is present versus if it's not present, that there is essentially no response to the TLR4 agonists or LPS and the knockouts. When you re-express it in male sensory neurons, you get some effects, but then in the wild type animals where everybody has TLR4, there's a pretty major effect of toll-like receptor 4 mediated IL-6. Looking in females, what we found is a couple of things. One, kind of difference in the y-axis, but when we reactivated TLR4 in the peripheral sensory neurons and compared to wild type animals, they're right on par. So it seems as if the TLR4 mediated IL-6 is coming from peripheral sensory neurons in females at least. And then looking at the whole body knockouts, there's pretty much nothing happening going on. And the vehicle groups for good measure. And then we get really excited because we're like, oh, maybe this is the same thing that's happening with the CGRP response. And so we took, waited a few hours less and did the same experiment to pull out CGRP. And we saw, while there is obviously LPS effects of CGRP release, we didn't see that stark difference with reactivating TLR4 in the sensory neurons in the males versus the females as we did before. So it's not as home run hitting as we want, but we do think that there's definitely an effect, and it's sex leaning. And so we're kind of delving a little bit deeper and trying to figure out more questions or more answers to the questions that we're making for ourselves. So some concluding thoughts about all of this is that nociceptors mediate sexual dimorphic responses to TLR4 agonists, probably other things as well. Macrophages tend to be a part of the conversation in long-term plastic changes in males versus in females, it seems to be nociceptor mediated in the context of TLR4. And this alludes to this really interesting dichotomy where if everybody's in the pool at the same time, you don't see huge differences, but there's clearly this neuroimmune interaction, neuroimmune crosstalk between the different cell types to get you to the endpoint. And there's been a couple of papers that have kind of looked into that, looking at transcriptomic sex differences in peripheral sensory neuron populations, as well as transcriptomically, as well as proteomically as well. Cool. And so this is a picture of my lab from a few years ago. And here goes all of our collaborators, which quite a bit, and then some funding sources. But thank you all for your attention. All right. Oh, got a hand clap online. All right, let's open it up. Yes. Yeah, so the question was about transferring macrophages from the males of activated males to females. We have thought about it. We haven't done it just yet. We've been just trying to really wrap our head around if there's any kind of clear mechanism that we can kind of pull out. But no, we haven't done it. But if I were to think about it and to kind of guess what would happen is we would see activation and sensitization. If I had to guess, but I'm just guessing. Don't get mad at me. Yeah.
So this is a really beautiful interrogation of that first sensory neuron. I was wondering how you thought about further on thalamus, EPM.
Yeah, not at all. And so, because you get into the weeds so fast, and we have a saying in my lab about rabbit holes. and try to avoid them. But it's super hard not to slip into one and fall, incredibly hard and deep. But we haven't really thought about it a ton, especially at the level of the brain. We have thought about it in the context of the spinal cord, local levels within the dorsal horn, microglia activation, and we have some interesting stories looking at sex differences in the spinal cord. So, cool.
Other online questions? Do we have to check? I think they show up in the chat for the webinar.
Yeah. So no questions that I can see, but yeah, go ahead. No, so the knockout is what we consider that, but I know exactly what you mean. We haven't done it because we've been really focused on removing it and seeing what happens. But yeah, it would be really interesting. There's a group at San Diego that has quite a few of those types of experiments. TAC242, I think that's what it's called, and it's a TLR4 pathway inhibitor, and there's quite a few cool studies that they've done out there. So cool. Yes. Yeah, I think people can hear that, right? Did we expect to see that? No, actually, the original hypothesis was that neurons would do X and macrophages would do Y, but there wouldn't be a real sex effect. And that's because, well, toll-like receptor 4, it to us seemed fairly redundant in the context of where it was expressed because we didn't see the sex differences. Like when I said that, I saw people going, right, and it just made sense. Like it probably wouldn't do anything, but it did. And I think what I would say is making sure that we approach these particular kinds of experiments properly. And the biggest thing is properly powering them. Because you read a ton of papers now that say, oh yeah, we looked, but we didn't see anything. But then here's our N6 with all of our animals together. And you're like, well, yeah, 6 animals is probably enough to do some statistics, but not to do the sex difference comparisons. And that happens way too much. And so that's what I would just say, make sure that you have to think about the proper power. It's really rough though, right? Because I mean, now experiments are double the size because you have to do that, but or at least 30 to 50% the size because you have to do that, but it's super imperative too. So yes. Ooh, my back hurts today. Yeah. Yeah. So we are super interested in aging. However, we haven't done the combined aging and transgenic experiments. However, we do see sex differences in aging that are super weird. And I don't know how to explain it. And I can't explain it good enough to get a grant to do more of it. And I can't, and folks just don't want to give it to us. But so what we find is that, especially in males, the robust effect of like a post-operative pain condition or an inflammatory pain condition, that they're actually not in as much pain or severe as females. And it's been kind of recapitulated in the clinic if you take out data and you superimpose it and flip it around and look at it different ways. And a lot of people have said, oh, there's no differences, but because it wasn't powered enough. But when you actually do an experiment and you try to power it, we see that, and we keep seeing it over and over again. And other people have seen it as well. So there's, while we haven't done the TLR4 necessarily approach, what we do just see age does have an effect. With females, though, it doesn't seem to really be different, and at least in the context of the onset of a pain state and the recovery from one. But in the males, there seems to be this delay in max pain state and nowhere near as painful of a state. And there's been a couple people that have been publishing some really cool stuff recently about that. Okay. Yeah, super interesting. Yeah, we connected with the neurosurgeon team at UT Southwestern. They're spinal correction surgeries, so it's not exactly the same, but the same, right? And most of these individuals are 50 plus, and so aging is like superimposed on top of it all too. So yeah. Overectomized females in this model, we haven't done that, no. We have tracked some estrous cycle to see if that has some effect across time, and it doesn't seem to, but I would argue that that's not powered enough. We only did it a couple times back in the day, but no, we haven't done it either. So what hormones are doing that, if any at all? And it's hard to tell, and I think. Yeah, I have a thought on that. And I guess I'll share it. I think essentially it's hardwired early on, and that fluctuation of hormones. What's hardwired the differences early on, but the fluctuation that we're finding or that we see with hormone changes across cycle are just too fast to really have that kind of influence. And so, and people have been showing mostly no differences across the board when they look at that, and it's probably angering the people. We definitely still need to keep doing it though. So. Ooh, fancy.
That's like sufficiency in the home.
Yeah, cool. All right.
Thank you very much. So, wait, pizza, of course, there's not pizza available for the people on Zoom. I guess we can serve them virtual pizza.
Sorry, no pizza, y'all.
Thank you very much.
Awesome. Well, thank y'all.